That's fine.
So hello everyone and welcome. I'll just give people a few moments to get settled. Okay, so I think we are good to go. So thank you everyone for joining. It's great to have you here and hopefully you're able to enjoy today's presentation. So my name is Caitlin Jaggers. I am the research lead at the Alzheimer Society of Canada, and I work within the Research Knowledge Translation and Exchange team. At the Alzheimer Society of Canada, we're very pleased to welcome you to another ASRP Exchange presentation that is being offered through our provincial support program. This series has been developed to feature Alzheimer Society research program recipients who have completed their funding and who will speak to the outcomes of their ASRP supported research while addressing how their results have and will continue to impact people with lived experience. Joining us today is Sarah Sarovich, the Research Program Assistant at the Alzheimer Society of Canada. Sarah will provide a brief introduction for our presenter and walk us through today's agenda. We also have Erin Crawford from the Alzheimer Society of Manitoba, and Erin will be moderating the question and answer period following today's presentation.
Hello, everyone, and welcome. As Caitlin mentioned, my name is Sarah Sarovich, and I am the Research Program Assistant at the Alzheimer Society of Canada. I would like to start today's webinar by going through the agenda presented here, and then I will pass it off to our presenter, Dr. Michael Jackson. Before we begin, please note this webinar is estimated to be under 1 hour in length and is being recorded, but that only the video and audio of the presenters is captured during the recording. The presentation recording, in addition to the PowerPoint slides in PDF form, will be made available on Connection for Alzheimer's Society staff, as well as on the Alzheimer's Society of Canada's website on the ASRP Exchange webpage following today's webinar. Today's webinar will be presented by Dr. Michael Jackson and is being offered as one of the ASRP Exchange webinars within our provincial support program, Meet the Researcher series. We are so pleased to have Dr. Jackson here with us today to present his project, Identifying Prospective Therapeutic Targets in Alzheimer's Disease, Understanding the Cascading Effects of Cellular Deterioration. This project was funded by the Alzheimer's Society Research Program, the ASRP, starting in September 2018, with project completion ending September 30th, 2021, which is tomorrow. Dr. Jackson joined the Department of Pharmacology and Therapeutics at the University of Manitoba in 2013. His laboratory is in the Neurosciences Research Laboratories in the Clayson Institute for Advanced Medicine. And Dr. Jackson obtained his PhD from McGill University in the Department of Pharmacology. Following postdoctoral training at the University of Toronto in the Department of Physiology, he moved to the Robarts Research Institute at Western University in 2008 as a research scientist and adjunct professor in the Department of Physiology and Pharmacology. A major focus of Dr. Jackson's research is to understand how families of calcium ion permeable channels contribute to calcium ion dysregulation, impaired synaptic transmission, and plasticity, as well as neuronal cell death in models of stroke and Alzheimer's disease. This focus follows from a body of work demonstrating that calcium ion permeable channels are functionally coupled to the NMDA glutamate receptor subtype and contribute to physiological and pathological processes. A primary aim of this research is to gain mechanistic insight into how these calcium ion influx pathways are regulated by intracellular signaling pathways with an overarching goal of identifying molecular targets for development of agents effective in treating neurodegenerative disorders. As mentioned, at the end of today's presentation, Erin Crawford will be moderating a short question and answer period. Please use the Zoom chat box feature if you have any comments you would like to make or questions that you would like to pose during the presentation. Please note that we will hold off on asking our presenter to respond until after he has completed his presentation. And without further ado, please join me in welcoming Dr. Michael Jackson.
Thanks so much, Sarah, for that introduction. So let me just go ahead and share my screen. So hopefully you can still see the screen and everything is good. Perfect. Okay. I do want to thank everyone for the introduction, Sarah for the introduction, and Caitlin for the invitation. So it's a great pleasure to come and talk to you today about a summary of the research that we've been doing that was funded by the Alzheimer Society. It's interesting, Sarah mentioned that the funds are actually coming to an end. I didn't put that together, so the project has come to an end, but the research does continue, and I'll highlight that as I get towards the end of my talk. So the intro was actually useful also in providing a little bit of context. So I recognize that the audience may not have a scientific background, so I've tried to bring the topic in a more approachable manner. But as identified by the title in the introduction, really my lab is focused on identifying mechanisms of disease, which could be potentially exploited in developing novel therapeutics, which are, of course, greatly needed in Alzheimer's disease. So the first slide I wanted to show was just, again, to thank the Alzheimer's Society for funding and also the donors who give to the Alzheimer's Society. The title of the proposal that was submitted to the Society is shown here. And I also wanted to acknowledge that this is a collaboration with my colleague, Tina Kaupanen, who was a co-applicant on that proposal. And we continue to collaborate. She's actually just a couple doors away from me. So we're in the same institute, the same Department of Pharmacology at University of Manitoba. Tina's area is really neuroinflammation. This is an area that I've moved into more recently in collaboration with her. And so what I want to talk about is our research, which has moved in this direction to look at targeting neuroinflammation in Alzheimer's disease. Now, before getting into the specifics of my research, I just wanted to highlight some of what we know about Alzheimer's disease. So I think everyone in the audience recognizes that Alzheimer's disease is the most common neurodegenerative disease, which is estimated to affect over 20 million cases worldwide. Compounding the burden of disease are a number of alarming facts. So this is an infographic from actually data from the Alzheimer's Society dated 2011. So what we see is that at the time it was estimated that approximately 750,000 individuals in Canada are afflicted by Alzheimer's disease. And what is alarming is the fact that it's expected that the prevalence will increase with aging population. So we know, of course, that probably the major risk factor of Alzheimer's is aging. And so as the population ages, the incidence doesn't change, but the number of people afflicted will increase. So it's estimated by 2031, there would be approximately 1.4 million individuals affected. So this is a doubling. And we can see that the economic cost associated with dementia would increase tenfold and would balloon to somewhere in the range of 293 billion. Of course, these are just estimates, but it just shows the burden of a disease that may be pending. Contrast that with funding from CIHR, which is the main body for funding in Canada, which represents only $236 million as of 2011. So this really illustrates the need for more research into this area. This graphic also illustrates the fact that we know that women are more commonly affected by Alzheimer's disease, but there's also evidence to suggest that in some cases, the outcomes in men may be more severe. So there's an important sex difference in Alzheimer's disease, which is important to study in the future. Equally alarming is the fact that there are no treatments available to alter the course of disease. So listed here are drugs that are currently approved for treatment of the cognitive symptoms in Alzheimer's disease, and Abixa and Aricept are maybe the most recognizable. I believe they are approved here in Canada. They were approved since 2004. They target two distinct mechanisms that are related to learning and memory. In some cases, these drugs may be used in combination, but it's important to note that irrespectively, we know that these drugs have very modest benefit in alleviating symptoms, and they certainly don't alter the course of disease in patients. So there is really an urgent need to do more research and identify new mechanisms of disease which can be targeted for new therapeutics. Now, with that in mind, it's important to recognize that Alzheimer's disease, of course, is a progressive degenerative disease. So in the late onset form, which is much more common, we know that the onset of diagnosis is typically in the range of 65 to 70 years of age. Afterwards, progression is very rapid, and death is always the end outcome of Alzheimer's disease in patients. But it's important to recognize, and we do know that the molecular changes that are at the root cause of Alzheimer's disease occur many, many decades before the first symptoms emerge. So the mildest form would be amnestic MCI, or mild cognitive impairment. And so we know that even before the first symptoms occur, the pathology has already set its course. Now, this is alarming, but also comforting in a way, because there was a study published in 2007 that identified that any treatment able to delay symptoms by as little as 1 year would be able to reduce global prevalence of disease by over 9 million cases over a 4-year period. So there is hope that if we can find not necessarily a cure, but any therapy that could delay the onset of symptoms, patients would be, the prevalence of disease would be largely reduced. So with that in mind, again, the goal of my research program in collaboration with TNET is to identify molecular mechanisms that may be exploited to develop new therapeutics that would target these disease-causing mechanisms. So before getting to that, I wanted to just summarize a little bit more what we know about Alzheimer's disease pathology. So shown here is an Alzheimer's brain at autopsy. And it's quite evident that there's severe atrophy, shrinkage of the brain. So this can be seen in different regions, shown here, the cerebral cortex. Also shown here and important is that Alzheimer's disease will affect the hippocampus. Now, I'll talk a little bit more about this region of the brain. It's very important for learning and memory, and one of the first areas to degenerate in Alzheimer's disease. Now, if we look at this by histological examination, we see the characteristic lesions, which are the hallmark of an Alzheimer's brain at autopsy. And these are the amyloid plaques and neurofibrillary tangles. So the amyloid plaques were first identified over 100 years ago. But it's important to recognize that amyloid beta, which I'll talk more about, is the protein that was isolated from these plaques. And this only occurred in 1985. So this is really the start of the modern era of Alzheimer's disease research. Now, in the intervening years, there's been a number of studies that have gone on to show that amyloid beta is acutely toxic to the brain. More specifically, it's toxic to neurons. So I think everyone recognizes that the brain, the human brain, is estimated to have approximately 1 trillion neurons. And all of these neurons communicate with one another through a very fine structure, which is referred to as a synapse. So synapses are on the scale of 1/1000th the size of a human hair. And what we now know is that amyloid beta is in fact quite toxic to the synapse. So long before the neurons die, some of the first manifestations of disease may be due to the effect of amyloid on synaptic communication, in particular, soluble forms of amyloid. So there's a lot of focus in the past on these plaques, but what we now know is that the correlation between cognitive impairment and plaque burden is quite poor. More relevant is likely the accumulation of amyloid in its soluble form, which again is synaptotoxic. So it impairs communication at the level of the synapse. Now, in my lab, we focus on the hippocampus for reasons that I mentioned earlier. It's one of the areas that are very important for human memory. So episodic memory in particular. So that's the ability to recall day-to-day events in our lives. And we also know that this structure is preserved in all mammals. It's important in rodents, for example, in recognition memory, which is something that we can assay in our experimental settings. As I mentioned, it's one of the first areas in the brain to degenerate in Alzheimer's disease. And it's also an area for which is the foundation of studies looking into mechanisms of neuroplasticity. So when we talk about neuroplasticity, so this is a change in the strength of communication at the level of synapses. So this is the way in which neurons communicate with one another. And neuroplasticity broadly refers to our ability to adapt to changes in our environment. And it's the foundation of learning and memory in humans as well as in all mammals. So neuroplasticity is critically important. And the way we study this typically is shown here. So what we're able to do is take the hippocampus from a rodent, so this could be a mouse or a rat, and we can take thin sections of this area of the brain acutely and maintain them in vitro setting. So here we have complete control over the environment. So we're able to position recording and stimulating electrodes, and we can record the synaptic communication between neurons. And an example is shown here. So if we apply electrical stimulation, what we can record is a synaptic response. So again, this is the communication between neurons. And if we record this at a low basal stimulation rate, so let's say 1 every 30 seconds, what we find is that these responses are very stable. So shown here is 20 minutes, but we can record these for many, many hours and the response amplitude doesn't change whatsoever. So the fidelity and strength of communication between neurons is maintained. And what we know is that plasticity is induced by change in activity. So here we do this experimentally by applying a high frequency train of stimuli. And what occurs then is an immediate increase in the strength of communication between neurons. So this is seen as an increase in synaptic strength, which is the very definition of an increase in plasticity. So think of this as being a foundation. We know that this is a foundation for learning and memory. So where this is important when thinking about mechanisms that cause loss of learning and memory in Alzheimer's disease is shown here. So what we're able to do experimentally is that we can treat these slices with amyloid beta. So this is soluble oligomers that we synthesize in the lab. And we can immediately see that treatment with amyloid beta is able to impair this form of plasticity that we record in these slices from the hippocampus. Now, this is a concentration dependent effect. So if we increase the concentration of amyloid, we can completely abolish this response entirely. So this is very likely the foundation of impaired cognitive function in Alzheimer's disease patients. So all of what I've mentioned earlier until now forms the basis for the amyloid cascade hypothesis, which was first formulated in 1992. Largely formulated on the basis of plaques that I mentioned earlier, but more recently revised to include the occurrence of these soluble oligomers that are synaptotoxins in impaired neuronal communication. So what's important to recognize is that this amyloid hypothesis led to all of the strategies that have been attempted to date in terms of clinical trials. So all of the drugs that have been attempted have attempted to inhibit the production or the clearance or increase the clearance of amyloid beta. So shown here is amyloid beta in a monomeric soluble form. And we know that it's generated through the enzymatic processing of a precursor protein, appropriately called the amyloid precursor protein. So monomers are first synthesized, as I mentioned, and what's important to note is that these have a propensity to clump up together, to form aggregates. So this is shown here as oligomers, and eventually these oligomers deposit in the form of amyloid plaques. And these amyloid plaques are then thought to induce changes in tau, which is a linked pathology in Alzheimer's disease. So as everyone likely recognizes that to date, the mechanisms that have been attempted in trials targeted either the enzymes that lead to the production or used immune therapy to try to increase the clearance of amyloid beta. And as you well know, all of these strategies failed clinical trial. And typically the most common reason is either a lack of efficacy or toxicity. So this is important to keep in mind. So toxicity, at the time that these trials were first initiated, it wasn't appreciated that these enzymes also serve other physiological functions. So they not only process amyloid precursor protein, but they process other proteins which are important in the brain. So this could be one reason for toxicity. A reason for the lack of efficacy is that amyloid beta is acutely very, very potent. So I'm not convinced from many of the trials that have been attempted to date that they've been able to clear enough of the amyloid at sites where they cause injury and at an appropriate time. So ideally, this should not be applied in severely demented individuals, but ideally, any treatment would be targeted at a much earlier time when amyloid is starting to accumulate. So this could be major reasons why this has failed, and it really doesn't encourage further trials into drugs that would clear amyloid beta. So rather, it really highlights the need for new therapeutics that target disease-causing mechanisms. And for this, it's important to recognize, again, the effects of amyloid beta in promoting impaired cognitive function. So I mentioned earlier that our research in collaboration with TEEN has been focused on neuroinflammation, and I wanted to highlight a little bit about the inflammatory theory of Alzheimer's disease. So the first indication that inflammation was relevant came from correlative evidence, so these would be clinical trials. that seemed to indicate that patients that were on long-term anti-inflammatory drugs, so think of individuals taking NSAIDs, for example, ibuprofen for arthritic pain, it seemed to be a reduced risk of Alzheimer's disease in these individuals. So this is perhaps a little bit controversial. There's certainly evidence to support that, but what I would point out is that probably NSAIDs are not the ideal drug for targeting inflammation in the brain. I would point to more convincing studies from genomic studies. So we're in the genomic era. And so this includes genome-wide association studies that have identified variants that are linked to an increased risk of Alzheimer's disease in individuals with late onset. So this is not the acute familial. This is the more general sporadic or late onset form of disease, which I mentioned occurs typically in individuals 65 years and older. What's also important to recognize is that many of these variants that have been identified are linked to genes that we understand play a role in inflammation. In particular, they seem to be highly expressed in microglia, which I'll talk more about, which are the immune cells in our brains. So how this inflammation theory works is shown here. So I already mentioned that amyloid beta, which can form plaques, is also acutely toxic to neurons. So I mentioned that it can cause loss of synaptic communication and ultimately lead to the death of neurons. Now, what we now know as well is that amyloid beta induces inflammatory stress. So typically in the brain of an Alzheimer's disease patient, there is chronic neuroinflammation, and inflammation is now considered a hallmark of an Alzheimer's disease brain in patients. Now, this inflammation, which is a complex response with many, many different components, can be induced by the death of neurons. So microglia, for example, respond to injury in the CNS, but can also be caused by amyloid beta stimulating a gain of toxic function in microglia. So I wanted to discuss this a little bit more. And this highlights how we might come about targeting chronic inflammation in Alzheimer's disease. So when we talk more specifically about microglia, I mentioned that these are the immune cells in the brain. So previously they were thought to either be in a resting state, but we now appreciate that even in their so-called resting state, they play an important role in surveying the brain and providing trophic support, so growth factors that would be beneficial in regulating function in the brain. Now, upon injury or invasion by a pathogen, microglia respond, and this is a physiological function, and they produce what's referred to as pro-inflammatory response or pro-inflammatory cytokines, which are intended to ward off toxin and terminate that toxic injury event. Now, following this, they revert to an anti-inflammatory mode where they promote repair and secrete trophic factors again to try to restore normal function of the brain. So I've listed these as bad and good. And what we now know is that in Alzheimer's disease, as I mentioned, amyloid beta is able to stimulate chronic neuroinflammation. So what this is associated with is a loss of the beneficial function of microglia. So they no longer provide this resting trophic support, and they adopt a pro-inflammatory toxic gain of function where they exacerbate injury by amyloid beta. And this leads to a spiraling cascade of events that ultimately compromise neuronal function. So this leads us to the target that I want to talk more about, which is shown here, which is the enzyme called PARP1. And what we know from pioneering work by my colleague Tina is that amyloid beta is able to stimulate increased activity of PARP. And the reason this is important is that PARP has been linked to increased inflammation, pro-inflammation in microglia. So what we know is that with PARP being activated, microglia lose their capacity for their beneficial response, and they primarily gain a toxic gain of function where they secrete a spectrum of cytokines, which are ultimately cause damage in the brain. So one strategy for alleviating this is to target PARP with inhibitors, as shown here, or genetically in our mouse models, we can simply delete the gene from microglia. And this is what's illustrated here. So I showed this response earlier where amyloid beta is able to suppress neuroplasticity. So this is from control mice. And what we see below, when we repeated this experiment in mice in which PARP1, this enzyme that I mentioned earlier, is deleted from microglia specifically, what we see is that amyloid beta loses the capacity to suppress synaptic plasticity. So this is very, very encouraging from a therapeutic perspective. And I should also point out that we know from animal studies that deleting PARP and microglia is also able to alleviate cognitive impairment in mice. So you might think that inhibition of PARP would be ideal, but what we know about PARP is that it's expressed throughout cells in our body and plays a very, very important role in maintaining what's referred to as genome stability. And so inhibiting PARP broadly is not the ideal target because of risks of side effects. And this is driven home by the fact that drugs that target PARP are primarily been developed as cancer chemotherapeutics. So they have a heavy number of side effects associated with their use. So this led us to explore in the research that was funded by the Alzheimer Society alternate means by which we can regulate PARP. And this is illustrated here. So this is the culmination of 3 years of research illustrated graphically in one slide. So what we were able to show in collaboration with Tina is that amyloid beta stimulates microglia to release the excitatory neurotransmitter glutamate, which acts on a glutamate receptor, which is also expressed in microglia. This initial activation of NMDA receptors causes a rise in intracellular calcium, which then stimulates the activity of PARP. So this is the initial insult. But what we went on to characterize is the relationship between PARP and another gene called TRPM2. So this is a protein whose highest expression is in microglia. And what we were able to show is that TRPM2 is absolutely essential for maintaining PARP in its constitutively active form, which drives increased chronic neuroinflammation. So what's important to know is that PARP also itself activates TRPM2. So what we have here is a vicious feedback cycle, which promotes increasing PARP activity, which sustains microglia in this chronic pro-inflammatory mode. And so what we're now pursuing are strategies to eliminate TRPM2 activity in microglia. And so the final slide I wanted to show is shown here. So what we know about TRPM2 from my own separate studies is that TRPM2 is also expressed highly in neurons. And we've been able to show that amyloid beta stimulates TRPM2 expressed in neurons to cause neuronal injury. Now, separate groups have also shown that TRPM2 is highly expressed in endothelial cells that form the basis for the blood-brain barrier. And they show that amyloid beta is separately able to cause changes in the blood-brain barrier, which promotes increased permeability, which is ultimately toxic to neurons as well. So one of the future directions with funding from CIHR that we received recently is in collaboration with Tina to explore more in-depth using sophisticated genetic models, whether TRPM2 expressing these different compartments is primarily responsible for the toxic effects of amyloid beta. And also important in terms of drug discovery efforts, we initiated collaborations with groups out of University of Toronto, so Dr. Jim Eubanks as well as Mark Reed, to look at a screening of compounds, a library of compounds, to identify TRPM2 inhibitors, which would have favorable blood-brain permeability profile, so meaning they would be accessible to the central nervous system. And what we predict based on what we know is that inhibition of TRPM2 in any of these compartments would actually be beneficial towards restoring cognitive function. Lastly, very recently, we initiated a collaboration with a group out of the University of British Columbia, Brent Page, who contacted us with his results that show that a separate gene called NUD-T5 unexpectedly is also linked to the cascade that I mentioned earlier. So he is a medicinal chemist who's identified very specific inhibitors for this enzyme, and he's very motivated to study whether these inhibitors may provide another means of targeting this cascade that I mentioned earlier, which would be able to relieve the chronic inflammation in Alzheimer's disease. So with that, I want to turn to my last slide and acknowledge all of the team members, both past and present. So highlighted in red are members of my lab, also members that are jointly supervised with Tina, members both at the University of Manitoba as well as from Western University, where we were located previously. And also I want to give a nod to our collaborators, both at Western University as well as University of Toronto and UBC. And lastly, again, to thank the Alzheimer Society for their support and now ongoing support by the Canadian Institutes for Health Research. And so with that, I'm happy to answer any questions that you might have. Thank you.
Excellent. Thank you, Dr. Jackson. That was a very interesting and informative presentation. I think you were able to address an area that might not be well known by a lot of general public members. I think, as you said, this new era of the amyloid beta research is definitely on people's radars, but there's so much else that needs to be explored in that area. And I think you really targeted a specific area that is of a lot of interest to people. So now is the opportunity, as Dr. Jackson mentioned, to pose any questions. If you do have any, I'm speaking directly to our audience members at this time. So you can do so, as Sarah stated at the beginning of the webinar, through the Q&A box or the chat box. Either function is available to you. I know that some people take some time just to digest information and maybe want to just reflect a little bit. So maybe while the audience gathers their thoughts and has some time to think about some questions, I do have some questions myself that I can pose in the meantime.
I do too, that I came up with while he was.
Oh, perfect.
Talking, actually.
Oh, that's great. Thanks, Sarah. And so I'll maybe just ask one or two, and then I can pass it over to you, and we'll also keep an eye on the audience box to see if any questions are coming through. So I think kind of a broad question, but I'd like to know if there's one thing, Dr. Jackson, you would like the audience members to take away or remember about your research. What would you like that to be?
I think it's really just maybe encouragement or a cause for optimism. I think the field of Alzheimer's has been fraught with all these negative results from trials, which has been very demoralizing. But certainly what I feel is that we're in a new era of research. I pointed to the genomic studies, which really shine a light on some important targets that have been maybe previously underappreciated. So I think if there was one point I'd like to take home is that we sense a renewed sense of encouragement in driving these studies and identifying molecular targets. And these are important simply because ultimately the drugs that would be developed as therapeutics ultimately target molecules. And so identifying these important molecules that are driving disease is really, really critically important.
That's great. Thank you. Yeah. And certainly I think some individuals may feel disheartened with certain results or certain things that are released in news and other platforms. So it's definitely important to hear research that inspires hope and encouragement about ways forward. Maybe I'll pause and I'll pass it to you, Erin, if you have a question as well.
Yeah, partly question, partly comment. My comment is more, for me, I'm so happy to be part of an organization that is fundraising for this kind of research. But I want to say thank you to you for having chosen to get involved in this kind of research, because I know it's a challenging field. I'm told it's a challenging field to be doing research in. And so to hear you, even for me, kind of the most exciting thing was when you said that out of the blue, a researcher from UBC reached out and said he was doing something that he thought related. And I know that even though the results in research generally haven't been as exciting as we would like or as promising for the clients that we talk to, that having that dynamic research community where somebody says, I'm doing this, I think they might connect, is how anything's going to happen. So I want to say thank you, and how exciting that comment was, even though it's in some ways just such a minor thing. And
maybe I can add,
yeah,
how important actually the Alzheimer's Society plays a really critical role for that. And I've mentioned that I've been happy to participate in reviewing grants as well. And this really forges a community of researchers. So maybe donors don't always appreciate that. When we come together to review grants, and I guess this itself is sometimes demoralizing because we hash through the science but I think we build bridges. We see the latest research that's being done, and it really fosters a sense of community, encourages people to stay in the field. And the funding is important, not only for the research, but also in training the future generation of researchers that are going to develop therapies in the future. Yes, yeah. Thanks for that comment.
I thought it was interesting to the comments on, or the focus on, inflammation, because I've heard lots about inflammation in other areas, but not with respect to Alzheimer's disease. And so it's in some ways really hip topic in various areas of disease prevention. But I guess being new to the idea of it here in Alzheimer's disease, like how promising do you see this as a new area? Is it just figuring out, like you think this is it and it's just figuring out how to get at it? Or is there still the potential that you as researchers go down this road and the door closes? Do you have any comment on that?
I think what I pointed to was the gene association studies. So this is really an emerging area. I think even 10 years ago, we wouldn't have been talking about this. And so there's something that I'm happy to talk about more, but it's not my area of expertise, but there's something that's being refined called the polygenic risk score. So each of us carry a number of variants and mutations in different risks. And this leads to the hope that there might be better identification of individuals that are at risk. So that's important in terms of maybe identifying people who could benefit from treatment. So I know the field broadly is highly, you could, if you're a pessimist, you can look to all these studies that have failed and think there's no hope. But there are a number of limitations in terms of clinical trials. So oftentimes patients who are with severe disease are really willing to try experimental reagents, understandably. But I think there's a view that probably maybe the trauma and injury is too severe. So it's maybe unlikely that could be reversed in these individuals. So diagnostic coming from genetic risk may be one element that's very important. And then maybe related to more specifically to your question about inflammation, it's definitely linked from the genetic studies. And from all of the studies, we know that amyloid is able to cause directly inflammation and even indirectly through the injury that occurs on neurons. So the great challenge there is just how to identify a target which would allow us to have a selective effect. So I mentioned a little bit how microglia lose their function. So experimentally, this may be surprising, but you can entirely eliminate microglia in the brain of a mouse. And what we find there is that that's not desirable. And that points to the fact that microglia not only have this detrimental effect, but they have a beneficial response. So again, driving home the point that we have to have a targeted approach, which would target the pathology, but not the physiology. And that is not a trivial task. Mm-hmm. But I do think that with maybe, as you said, it's a hot topic. A lot of individuals are entering into this space. So I'm hopeful that our target would be a promising target, but it may be unlikely that there's such a thing as a magic bullet as well. I think that's a lot of the goal for a lot of research, but it may be that multiple therapies would be able to be used in combination. And perhaps an extension of the polygenic risk score is that all of us have a different response to drug treatment. And so maybe something in the future would be identifying both individuals at risk and also hopefully identifying therapies that may benefit them in particular. So I hope that answers your question.
Yeah, it's a really cool area for sure. Yeah. It looks to me like people are feeling shy today and not putting questions in the chat. But I think it's just fascinating, the stuff that you're doing, and I'm so glad that you're pursuing it.
Oh, thanks for that.
Thank you.
Yeah, I completely echo Aaron's comments. I also am grateful you gave that nod to the peer review process and the ability that it provides to researchers in the field of dementia to have those important discussions and kind of build a bridge amongst each other to make those connections in the field. I know it is still considered a bit of more of a niche area of research. And so, establishing those connections and having those important critical discussions, I'm so glad to hear that it's been beneficial for you. And I know that it's a process that we will obviously continue to pursue as we work through our competition and assess applications that are received. So thank you for that comment as well.
Yeah.
I do have another comment. Again, it's a bit more, or question, excuse me, it's a bit more broad again. But taking it back to people with lived experience and the clients that Alzheimer's societies across Canada serve, how would you say your research will impact people with lived experience either now or in the future?
The inflammation is something that perhaps we could be more aware of. How this would lead to an immediate impact on patients is less clear. But I think lifestyle choices are important. And I think maybe many people are aware of dietary risk. So I think we're more acutely aware of the gut-brain axis. And at some level that may sound trite, but I think there's truth to that. So I think the one thing that can have an immediate impact is for people to be more mindful of their particular risk. So not to dwell on it, but my own father passed away from Alzheimer's. And so we know that individuals that have had a family member with Alzheimer's are at 30% increased risk of disease. So I think if you're aware of that, I think it's affected me in a way. I think I've been more mindful of my lifestyle choices. So I think you can make dietary changes, and you can make increase your exercise, for example. So that may not help people who are experiencing severe dementia right now, but maybe it's more hopeful for people who are family members that may be concerned about their own risk of developing Alzheimer's in the future. And certainly further afield, I really hope that the research that's currently underway will lead to new therapies. So maybe most scientists have to be optimists. I think there's a lot of barriers and difficulties in research. So I don't want to discount how hard the road ahead is, but I am optimistic. To be truthful, I'm optimistic that in my lifetime, there'll be something that'll be effective in providing relief. And again, it's not so much that we need a cure, although that would be ideal. I think we need just something that would delay the onset of symptoms in patients.
Thank you for that, Dr. Jackson. And I think, you've raised an important point that we do get a lot of questions and queries about from clients, and that is risk and how people can better prepare themselves, or what activities they can engage in to delay symptoms or just take care of their brain health. That's certainly a hot topic. And absolutely, it's important to be optimistic. Sometimes it can be challenging, but certainly optimism is what leads to discovery. It's what leads to progress. And sometimes it can feel like a long journey, but it's individuals like yourself and other researchers in the field that have gotten us to where we are so far and will continue to propel us forward. So I greatly appreciate those comments from you as well. This is more of a researcher targeted question. So some audience members may be researchers themselves, maybe pursuing current careers in research or planning to. So one thing that we've asked some of our presenters is if you have any advice for other researchers pursuing projects during the pandemic, and any challenges this may have posed, or if you have any, as said, any advice for individuals who may be going through similar challenges.
That's a good question. Yeah, I guess in the spring of last year, everything across the country ground to a halt. I guess we could say worldwide. So that was initially very worrisome and discouraging. But thankfully, at least in our case, we've been able to resume research activities. So we are under some restrictions, but we are fortunate to be in a closed facility where we're a limited number of individuals. Everyone is vaccinated. That's a mandate at the university. So we've slowly been able to resume activity. So I think, again, I keep saying optimism. I think it's sort of not being too discouraged. This is affecting everyone. I think there is great sympathy within the community. So I know we are being reminded as reviewers when we review not only grant application, but studentship applications to be aware of the restrictions that have been imposed by the pandemic. And I think I always marvel at the creativity of researchers. So I think people have been really adept at switching gears and finding a way to move in a different direction with what they're able to do at the time. So I think that kind of flexibility maybe is a good suggestion for people. So you may not be able to do what you intended to do, but in my group, we tried in the capacity that we're able to do some data mining to try to look at mutations within the human population for genes that are of interest to us. So there's always sometimes a way to find a way forward, even though when there's increasingly a number of barriers that prevent us from doing the work that we intended.
Thank you. That's excellent advice. And as you've mentioned, research, but the entire world has been impacted. And it's just about trying to find new strategies and work towards flexibility and looking towards potential new options. As you've said, there's even, this webinar is an example. There's options to meet virtually if you're, conducting a study where you interview participants, or, other means that can be utilized. It just sometimes takes a bit of time and reflection and the opportunity to just figure out what works best for you, and then utilize that time as best you can when there are those restrictions in place. So we're certainly glad that you were able to continue with your research during the pandemic. And as Sarah said, your funding concludes tomorrow. So, you powered through it even through challenging times. So that's definitely a nod to hoping for innovation and creativity and flexibility during challenging times. So
on that note, also, I appreciate, I mean, that the Society was gracious in extending the timeline for our funding. So that was a big help as well. So, and I think that was across the board. I think different agencies, different communities understood what was going on and supported researchers in allowing us a path forward. So that's been incredibly helpful. And again, to point to how important the Alzheimer's Society support was, it was really the foundation for what was to follow as our application to the CIHR, with which we were able to secure funding for another 5 years to drive this research forward. So that's been a great support.
That's wonderful to hear. We're so pleased to have been able to support you in this work, and it's excellent news that it's continuing forward and that there's plans for further delving into your research topic and hopefully expanding your findings. And it's all very exciting. Again, it's a challenging road, but it's definitely an important one, and we're glad that you're traveling on it. So as Erin said, it seems that there are not a lot of questions popping up from the audience at this time. So that's okay. Sometimes that's the case. People just are reflecting and digesting everything, so not a problem. So I think we can wrap up today's presentation a little bit early, and thank Dr. Jackson again for your time to go through your findings and to answer the questions that we've posed to you. Thanks again to Sarah and Erin for your time as well in putting this presentation together. I will note that should you have any questions that you would like to pose maybe after the fact or related to the webinar series in general, you can email psp@alzheimer.ca, and PSP just stands for the Provincial Support Program, in case you're not aware. And this actually wraps up our first round of ASRP Exchange webinars that began in October of 2020 and up to the end of September 2021. So thank you, Dr. Jackson, for kind of tying this off. Just a note to any individuals who are interested in continuing to attend these Alzheimer's Society Exchange webinars, we're just currently in the process of reworking a few things and kind of redesigning and making some adjustments, to the series. So please stay tuned. They are to come, and a schedule will be developed and released as soon as available. So just hang tight, and we will announce that news when it's ready. But for the time being, thank you for your attendance today. Thank you for everyone's collaboration, and I hope everyone has a great rest of the day.
Thanks, everyone.
Great, thank you.
Bye.